Hi viewers, in this class we are going to discuss the habituation. It is the one of the attitude of the learning process. Under this topic, we will see the introduction to the topic, definition of the habituation, experiments on say snail, experiments on the nereis, which is a polychaete, and habituation and ants on the honeybees, and finally the experiments on aplysia. Here, habituation is the simplest behavioral attitude of the learning process. The organisms in the habituation they lost their old significant responses. The stimulus, neither bound to reward nor bound to punishment, are repeatedly introduced to the organism. The organisms gradually stop their responses to the particular stimulus. In an experiment, while the snail is moving on a smooth glass plate, if you touch the plate, it withdraws its body into its shell. If you repeat this process for many times, finally it does not withdraw its body. That indicates it never responds to such stimuli. Here you are observing the snail. If it is if it is moving on a smooth glass plate, if you touch the glass plate, frequently it is withdrawing its body into the shell, and if you repeat it for many times, then it never responds to such stimuli, such shaking of the glass plate. If an organism is exposed repeatedly to physiologically insignificant stimulus, the natural ability to respond is gradually decreased and later on disappear completely in the organism. So, going on repeatedly, you are exposing a particular stimulus to an organism, so it keeping quiet without any responses. So that is it is habituating further situation. For example, there is a hunting dog. Upon the listening of the pistol sound, it disturbs and run away for some time. And if it listens daily the same sound repeatedly, so it results in the decrement of response of dog. So the dog do not show any kind of response after some days. That is, the dog loses its responsiveness to pistol sound. So it is habituating for the pistol sound. So this showing the behavior of the dog. If you observe some birds which are called as the gallinaceous birds, usually they see the cranes and ducks which have long necks and the short tails, so that they do not respond to the models with long neck and short tail. But if you inverse the model, that is, if you show the models with small neck and long tail, they are afraid of the models because of the inversion of the model. So that is the birds which are usually seeing which kind of birds, they do not show any kind of response. But if they see the inverse models, inverse structural birds, then they show some response. See here, they usually see the crane, ducks usually, which have the long neck and the short tail. If you inverse that, that is the short neck and the long tail, then they afraid of this kind of birds and as well as the models too. And there was a scientist named Clark. he made many experiments on the Nereis. Usually the Nereis is commonly called as a clamworm, which belongs to the polychaeta of the phylum Annelida. It lives in the burrows in the bottom of the sea, so that is showing the fossorial mode of life. In laboratory, he made a setup with a trough, and he arranged some glass tubes within the trough, and all these nereis were kept in the glass tubes. If you shake the trough, otherwise if you touch upon the heads of the nereis, or if you move any shade over the heads of the nereis, usually they withdraw their body into the glass tubes. The nereis, they withdraw their body into the glass tubes. If you do this repeatedly for 10 times with a time interval of 1 minute in between these repeated actions, then they habituated and gradually they do not withdrawing their body into the glass tubes. For 10 times they get habituated for that kind of situation. Here we are seeing the nereis. And if you give later on, if you give the electric shock for all these organisms of about 50% of the animals, they are the nereis, they are giving the response. And if the electric shock also giving electric shock, if it is repeated for 30 times, they slowly habituated for the electric shock also. During repetition of stimuli, if you decrease the time interval between exposure of the stimuli, then the habituation takes place very fast. Habituation takes place very fast. For example, if you expose the organism to a sparkling bright light with 30 seconds of interval in between successive exposures, the animals are habituated in 40 trials whenever the interval is 30 seconds. If you increase the interval for 5 minutes rather than the 30 seconds, then the animals for their habituation they require of about 80 trials or 80 exposures. That indicates that whenever the time interval in between the successive exposures is less, then the habituation is very faster. And whereas if you enhance the interval time, then they require more attempts, more exposures. The fastness of the habituation depend upon the nature of the stimulus also. Based upon the nature, the habituation is also changed. And if you provide any mechanical shock, shade, bright light, etc., these all are the different factors. They vary in their fastness of habituation based upon their peculiarities or specialities. Fastness of habituation changes with stimulus specificity. Based upon the stimulus specificity, there is a variness in the habituation phenomena. If you come to the arthropods, the nereis belongs to Annelida. And if you come to arthropods, habituation is a most common process in the animals of arthropods. If any organism enters into the hive of the honeybees or into the nest of the ants, they usually identify the organism by its smell and they kill the new organism immediately. If at all, the entire colony, if it is in the work, if you introduce a new organism, due to the work busy, they are unable to identify the new organism entry. Later on, they accept the new organism as one of the member of the colony, because all other organisms they are habituated for the smell of the new organism. So they took some time for the habituation of the smell of the organism. So this is the colony of the ants and the colony of the honeybees in a hive. Habituation is seen in all phyla right from protozoa to mammals in both invertebrates and the vertebrates. And moreover, habituation may be a fundamental feature of all living things, and habituation is very important in making adjustment to the environment through its behavior, through the behavior of the animals. If there are no consequences to a stimulus, for example, and whenever you expose this stimulus to the organism, then the organisms always keeping calm or keeping quiet without any response. It is also a correct decision sometimes, where there is no physiological importance and where there is no consequence with the stimulus, then ultimately organisms do not show any kind of response. So these all are the different animals right from protozoa to mammalia. And we will see the experiments of the Ganong. Habituation, he made experiments on in order to know whether the habituation is in the structural form or in the biochemical form in the nervous system. He decided to know it. And biochemical process of habituation are studied in detail in the Aplysia, which is a molluscan by the Ganong. And if you observe the Aplysia, there are of about 40% of the active regions in the habituation related nerve endings of the Aplysia. After getting habituated, the entire active regions are decreased to 10 or limited to 10. They are confined to 10% of the active regions. That is, whenever it is in the normal position, the active regions are 40%. After the habituation, they are only 10%. There is a significant decrease of certain ions in the nerve endings as well as endings of the receptor cells that elicit an appropriate response to that of the stimuli. And even muscle fatigue is not at all considered as a habituation, because the adaptive adjustment of the habituation is not at all seen in the process of muscle fatigue, so that we should not consider them as the habituation process. So this is the Aplysia, and here we are observing the nervous system of the Aplysia, where there are 40% of active regions are there in the normal organism. After the habituation, the percentage brought down to only 10%. And moreover, there is a significant decrease in the ionic strength, ionic concentration of certain ions in the system. That also indicates the habituation. This is all about the habituation and reasons, various examples related to the habituation. Thank you one and all. For more videos, you can watch and subscribe my YouTube channel, that is Swami Zoology. Thank you.